find something of value.
A higher education community in South Africa reflected on intellectualization.
How central this humanity is.
Welcome to The Academic Citizen. I'm your host, Mahita Ikani. This is our special season dedicated to the science and scientists linked to this SA Agulhas 2, our national scientific research vessel, on its 2025 voyage to Marion Island, one of the sub-Antarctic islands in South Africa's care. Marion Island houses a research base and sees a whole lot of science taking place 24/7, 365. If you haven't yet listened to episode 1, where you'll learn more about the ship as well as the geology and plant life on Marion Island, or episode 2, where you'll hear about insects, birds, and some very bad mice, I suggest you catch up on those first before listening to this episode, which is dedicated to the life in the waters around Marion Island, which is rich in biodiversity and hosts creatures great and small.
Normally in ecosystems where you have a lot of small things, you have much larger food webs. And with each transfer up the food web, the less energy is transferred up. Whereas if you have a few very large organisms, then you have a much more efficient transfer of energy up the ecosystem. So yeah, size matters.
You heard it here first. Meet Emma Swanepoel and Sarah Dupriya. They are both master's students in oceanography at the University of Cape Town and have joined the Marion Relief Voyage as research assistants to the oceanography team on the ship. They've both been sent by their professors to collect, label, and store specific samples of ocean water and associated critters collected by the CTD and plankton nets.
Very, 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 very fine scale mesh nets that are about 200 micron mesh. That's about almost a thousandth of a millimeter.
Remember the chief scientist, Marcel Vandenberg, explaining those in episode 1?
We've got 2 moorings that collect data continuously right through the year.
They're also very busy with their own studies, drawing on data that has been collected by other researchers on previous voyages.
The question is pretty simple. It's just, are the whale calls changing?
Each mooring has got an acoustic listening station used to pick up whale sounds and to identify throughout the year what whales is in this area and what season they do arrive.
A lot of people have studied the northern hemisphere fin whales, and a lot of people have studied Antarctic fin whales as well as Australian fin whales. And then when we looked at the study initially, they were pulsing at around 96, 97 hertz. That was in the initial data set, so 2014 to 2016. And they have since gone lower.
Why that is a matter of making well-educated guesses.
Figuring out why is very much less simple. I think it's important because vessel noise is bad for whales, and especially the low-frequency specialists suffer the most. Their calls don't travel as far as there's a lot of noise interference in a large ocean looking for other creatures.
But what do fin whales sound like?
Fin whales are low-frequency specialists, so they have super low pulses, usually around 20 hertz. There's downsweep from about 25 to maybe 17 hertz. And it's sometimes accompanied by an overtone in the 99 hertz range. And different fin whale populations have different frequencies at which they call. So interestingly enough, they're too low for us to hear. So humans can hear from 20 hertz to about 20,000 hertz. But obviously that diminishes from the day you're born. So generally, people only really start hearing from around 25 to 27 hertz to about 17,000 hertz. The only way in which you can confidently hear fin whale calls is if you speed it up, because that lifts the pitch higher. And literally all it is just like a like just a pulse. It's also really cool because when we study sperm whale calls or humpback whale calls, dolphins, orca, anything, the calls have a tone that changes. So you study the song in terms of how the pitch is changing in the time, versus in fin whale calls, if you're studying the actual song, you're looking at the spaces in between the calls, the silences, which in music is called the cadence, the beat between.
Okay, dear listeners, for 100 imaginary points, what do whales eat?
They're lunge feeders. They eat zooplankton, but it's usually krill.
Yeah, the slightly larger spectrum of plankton.
As you can tell, I interviewed Emma and Sarah together. There we were, sitting around the small table in the ship library with the microphone on the grippy mat.
It is quite ironic because you study the largest animal in the ocean and I study the smallest.
Yeah. Well, second largest and second smallest.
True. Yeah. You get micro as well.
Very cool.
But opposite ends of the spectrum for sure.
Yeah.
Sarah studies the plankton that whales eat. Her project aims to map the population of plankton in the sub-Antarctic waters around Marion.
You might wonder, why is it important to look at these tiny little microscopic organisms? But they actually have pretty enormous impacts on the ocean. Zooplankton are probably the most abundant form of life on this planet, which is a wild thought. And they make up the base of the entire food web.
Plankton are the most abundant animal species on the planet. Isn't that incredible?
So a lot of the animals that live at the Prince Edward Islands all rely on the zooplankton community as their main source of food. So if their source of food is changing or diminishing or getting smaller, that has a lot of knock-on effects to the predators at the islands as well.
So let's get back to basics. What is plankton?
Plankton in general are just all of the free-floating things in the ocean that cannot actively swim against currents. So they have an enormous size range, and it's not exclusively, you can see some of them with the naked eye. I think the smallest individuals that I look at are 1,000,000 times smaller than the largest ones I look at. The largest ones I look at being 20 millimeters. But they get a lot bigger than that, including jellyfish and things like that, because they aren't good swimmers. There's so many different types, and they're so different from one another. And almost alien looking, You get euphorsids, which kind of look like baby shrimp. These big worms, and we had this one that looked like a little Spanish dancer because it kept twirling and doing all these really beautiful swimming motions.
Sarah and her colleagues are studying zooplankton in the Southern Ocean because which plankton end up where can help oceanographers understand a lot about changes in the ocean.
Notoriously, the Southern Ocean is a very low productive environment because you have less sunlight during winter and you also have low iron concentrations, right? Which means that the phytoplankton are generally quite limited in their productivity. So yeah, these island ecosystems are really unique because they draw up iron from around the depths of the ocean, and you end up with these really productive oceans just in little hotspots around the island. So obviously you've got this huge uprising from the seafloor, which changes the currents around the island as well. And the Prince Edward Islands have a very unique bathymetry where it's got this very shallow inter-island shelf between the two islands, where the bathymetry goes from about 1,000 meters depth to about 200 to 40 meter depths very, very, very sharply. So all of those deep waters get washed up to the surface or around the islands. It changes the currents and the ocean very significantly. So you get these really extremely productive oceans around the islands due to the topography changes.
The climate of the planet is changing due to all the fossil fuels that have been burned since the start of the Industrial Revolution. And that is having a huge impact in the ocean, which is also changing from absorbing additional CO2.
We're seeing a lot of warming and a lot of southward shifts of these big major ocean bodies, which change the plankton that you see.
And this can be mapped by understanding what species of plankton are where. It might also be useful to understand what bathymetry is. Take it away, Sarah.
So bathymetry is basically the relief of the ocean floor. So if we think of mountains and valleys, basically the way we see land, the ocean floor is not flat. It has these same sort of features, even though we can't really see them and they're a bit of a foreign thought to most people. But yeah, you do get mountains and valleys in the ocean, and it changes the way that the water flows over them because it's either compressing or diverting the water column. and creating different like features within the current, which are very interesting. We're trying to basically understand what the variability looks like in the plankton in relation to the island and the different fronts that are present around the island, and how the ocean influences the variability that we see.
Zooplankton don't swim, they just get carried around by ocean currents. Before I spent time on the Agulhas too, I did not know what ocean fronts were. You might know what a cold front is, right, in the atmosphere, from listening to the weather report. But the ocean has fronts too.
So an ocean front is when two different bodies of water meet one another, and they generally have different densities. So one possibly being a warmer or higher salinity water mass, possibly from the north. So you've got like the warmer sub-Antarctic waters north of the islands here, and they meet the very cold fresh waters from Antarctica. And because these waters have different densities, they don't mix with one another and they push up against one another. And where they meet one another, it forms the front. And something very interesting about these fronts is that because you've got this tension between the two water masses, you get really fast-flowing currents along these fronts. So they end up being like quite an important factor when looking at the physical oceanography around the islands, because the position of the front determines the velocity of the current running through the island chain. So in years where you have the sub-Antarctic front closer to the islands, you have faster speeds pretty much pushing the water out of the island chain. And something very important with plankton in general is that you need quite slow moving water in order for it to heat up from the sun and form a nice little like warm sunlit layer where the phytoplankton can proliferate.
Right, so there is your crash course on the relation between ocean fronts and plankton. Remember, Sarah studies the second smallest kind of plankton out there. But what about the smallest? For that, we need to call in Dr. Zimkita Gebe, an oceanographer based at the Department of Forestry, Fisheries and the Environment.
I work in the environment aspect for oceans and coast, and I am a biological oceanographer within that unit, but I specialize in microbial ecology.
She got interested in plankton when she became curious about why fish stocks were depleting in certain parts of the ocean.
So I was an intern for the department for a year. And as I was there reading up on things and hearing people that have been in the department for so long, I heard that fish were depleting in the water. You would go on a cruise where they are supposed to collect or see more fish or like some nurseries of the type of fish that we were looking at. But people were complaining that there were no fish or there was little than it was in the previous years and whatnot. And so that sparked a question of what is happening in the water actually.
Dr. Gebe wrote her PhD on why fish stocks were depleting, and this led her to her current work on phytoplankton.
I study, I'm going to go a little bit scientific, autotrophic organisms as well as heterotrophic, which are the ones that do not source out any light, but then autotrophic are the ones that possess light by themselves, and they are usually called phytoplankton. But we also look at heterotrophic bacteria.
Remember, plankton are at the base of the food web. Everything in the ocean either eats plankton or eats something that eats plankton, or eats something that eats something that eats plankton. You get the idea.
I fell in love because after my PhD, instead of finding answers, I came out with more questions.
Dedicating her life to studying phytoplankton? Are you swooning? Because I am.
There are so many questions that still need answers. And so, yeah, that piques my interest.
Dr. Gebe explains that these very tiny organisms have not been studied enough yet in terms of how they use carbon dioxide and what their role is in the nitrogen cycle.
Most of the carbon dioxide and most of the nitrogen cycle that happens in the world is mostly done by the smaller organisms. So the size class that I look in is from 2 microns down to about 0.2 microns. But the larger organisms have been studied for years, and these biomass of microbes has not been looked at because there was no technology for them. So now it is important to actually come in and quantify how much we have of this and what their role is in the ocean in terms of time and also in terms of space.
A big challenge in studying these super teeny phytoplankton is that they are obviously too small to see with the naked eye and even with microscopes. And this is where some new tech makes things possible. How do we look at something that we can't see?
So I use a flow cytometer, which was previously used to count blood cells for years, but then they introduced it into ocean work as well. And so what it does is it has lasers that are set up inside of it. And so the water that I take here is only 2 mils out of the ocean. I only take 2 mils because in 2 mils of water, there are millions and millions of microbes. And so I put it in the flow cytometer and it sips it up. And as it sips it up, it sips each cell and it sorts them. So each cell goes up. It is hit by a laser. And then the autotrophic light that it has bounces back to the laser, and then it is counted according to what pigment it shows and what size it is. So what we do as part of our experiment is that we run it for 15 minutes at medium speed, and we try and get about 10,000 events. And 10,000 events is just the laser hitting a cell. It doesn't matter what kind of cell it is. It's just a laser hitting a cell, and then it gives us counts. And so that data then, I transfer it from the flow cytometer, which we use a BD Symphony A1. And then we take that and then put it on Flojo, which is a software now where you can subset and group those dots that you see on the flow cytometer into clusters of populations of the groups of microbes. And then we can calculate our abundances from those count numbers.
Okay, let's take a minute to digest all of that. Using a tool that was previously deployed in medical science, oceanographers like Dr. Gebe can now do cell counts on around 2 million organisms living in only 2 milliliters of ocean water and sort them by species, then calculate their abundance. How incredible is that?
Can't believe you came all this way to sample 2 milliliters
I know.
of water out of this vast ocean.
Yeah, so it's 2 milliliters, and then we replicate it 3 times.
So we're talking about 6 milliliters of ocean water and 12 million organisms. Mind blown. Those precious 6 milliliters come from the CTD. Remember our chief scientist, Marcel van den Berg? You can return to episode 1 for a refresher on the CTD if you need it.
When we do a station, the CTD goes down at 1 meter per second. So depending on the conditions, we get close to the bottom between 5 and 10 meters to be able to sample the whole water column. And things do change over depth. You get strong thermoclines, you get your plankton sitting in the upper layers of the water. You get different water masses that you can identify by all these parameters, depending how deep the casts are. If you go over a few thousand meters, you get various layers and how they affect the oceanography in any area that gets sampled. All the additional sensors that are attached to the CTD feeds data real time, and we can get fluorescence from one of the instruments, which gives us the amount of plankton that's in the water. The data gets fed back to us. And then from there, we can determine where to collect water samples.
At each sampling station, the CTD is sent down to pre-decided depths, and the bottles connected to it are closed to capture water from each of those depths.
We can close bottles by sending a signal down the cable.
I only choose 2 depths from the surface and from what we call a Fmax, which is the fluorescent maximum. We see where that is according to the CTD. As primary producers, most microbes sit more on the upper pelagic layer of the ocean. We try and take everything that is in the upper 5-meter layer. So sometimes your fluorescence maximum is at surface, and that means you only have 1 depth for that station.
And then once back on board, the scientists can then collect these samples at these selected depths for their experiments.
Imagine working on these kinds of scales. Milliliters of water and millions of organisms, counting their cells using laser and light. I guess this can all come across rather abstract, but remember that we're talking about phytoplankton here, which are eaten by zooplankton, which are eaten by all sorts of other creatures, including penguins and whales. So those tiny, tiny plankton are super important.
The story that I'm looking to actually tell is about the importance of taking care of the primary producers in the ocean. 70% of the world's oxygen comes from the ocean, and it comes from phytoplankton. Imagine a world where the atmosphere is just sitting there because of everything that's happening. If we take care and we listen to the people that are doing research in the ocean, then we know that if we close the system for a little bit of time, when we come back, it's going to recoup itself, because that's what life is. It recoups itself.
Yeah. Such a beautiful image of these microbes and phytoplankton like giving us oxygen and asking for nothing in return except to be left alone.
Just leave us for a little bit.
Okay, let's turn our attention now to creatures that we can absolutely definitely see with our very own human eyes. Maybe this makes us biased towards them. Probably. Other things that swim in the waters around Marion Island are penguins, seals, and orcas. Let's start with those charismatic black and white cetaceans, killer whales.
So I'll be documenting the orcas that visit the island and stay around the island all year. And then I'll also be tagging them.
That was Chandler Patel, a field assistant working with SAPRI, focusing on killer whales, who spoke to me on the voyage down. And this is Megan Clarkson, who did exactly the same job as Chandler until he took over. She spoke to me on the way back.
So mostly you're out there on the rock doing dedicated surveys, and each month it's different hours. So when you get there, winter starts with 2 3 hours and 1 8-hour a week, where you sit on the rock and you do a dedicated survey for that entire time looking for killer whales.
Yes, you heard that right. Megan and Chandler's office is a rock.
Kill Point,
Kill Point.
which is odd, but it's named after the killer whale. So it's a rock that's very close to the shore, where the whales also swim directly right close to the shore as well.
Once they come up, you take the time that you see them, which direction they're coming from, what are they doing.
So we'll be taking photos of their fins to identify the different individuals around the island, and using the markings and the notches that they have to know exactly who is who.
And then once they leave, you write down that time and anything interesting that happened during that watch. And if you can ID them in the field, you say who they were. So yeah, you get to know who they are, and they each have their own little personalities, what they do. And even sometimes you can see a behavior and you're like, I know who you are.
They spend daily shifts, no matter the weather, observing and logging orca sightings. Some days, they won't see any orca at all.
There were hours where you saw none for days. And you sit in all kinds of weather. Snow, hail, rain, wind. So you're hit with everything. And then obviously there are seasonal aspects to it as well. But mostly you can get up to, I think the highest we had was 70 knot winds. And yeah, I think the most I've walked in was about bordering on 60 knots. It like pushes you to the ground. You just have to get up and keep going. My first time seeing snow fall, actually, which was amazing. I think at one stage I had 9 layers on the top and 7 layers on the bottom. And yeah, you have your boots and your gloves. There's very few kinds of gloves which are waterproof that still give you the dexterity you need for a camera or writing your notes. Because you also take weather, and anytime your visibility changes or anything affects that sea state changes, you always note the time and write that in your data as well. I mean, I've had killers with me, which is amazing for like an hour, but you're in an hour in that weather with no gloves on. Your hands start to freeze.
It takes a special kind of dedication to do this kind of extreme whale watching. But remember that this isn't just for fun or curiosity. This was Megan's job for a year, and it's Chandler's job now.
When it comes to the biopsy sampling, it's done with a crossbow. Yes, so I've done my training. We're aiming on the saddle of the whale, so it's directly below the dorsal fin. And we're aiming to take around about 1 inch of tissue from there. So the crossbow bolt is fixed with a wire so that it can be pulled back. And with each shot, we're also measuring the whale's reactions to it. Because for some of them, they have a slight twitch. I was like, oh, like something touched me. They're not entirely sure what. For others, they have a bit of a freak out. So there's that. And then for the tagging, they have these geotags that are put on the dorsal fins that are only on for a short period, just a month. But it allows us to track their movements when they leave the island, and we can see exactly where these whales are traveling at different parts of the year.
In August, your 8-hour changes to a 10-hour. And that prepares you for the breeding seasons, which is from September to January the 15th. It's 2 10 hours and 2 3 hours every week that you're out there. And then winter goes back to the 2 3s and then the 8.
I'm not going to lie, I almost lost my mind when we had a sighting of 2 killer whales from the ship. My heart stopped for a full beat when I saw those tall, sharp fins cutting through the kelp.
So you're always scanning the water left to right in your whole field of vision all the time.
I'll just be watching and waiting and listening for when they come past.
Or you'll see a blow. And usually you see their fin first.
And those 6 seconds that day that the whales are actually there.
You scramble because you're so excited. You've been sitting for hours and then you note the time.
I'll just be snapping as many pictures as I can of all the different fins that I see. I need to estimate the size of the group that's coming by. If one individual is there, I need to be sure that I know that this individual might just be scouting ahead while the rest of the pod is further back and is going to be there in the next 20 minutes maybe. So the family groups I need to know, as well as knowing every single individual on the island now.
Most of the time they're inspecting the shoreline for food. So you'll see them kind of like going, they're not surfacing a lot and they'll be like cruising past under and you see them turn and they're lurking and they like inspecting the shoreline. And then, so that's most of the case. Sometimes then you'll see them surfacing quite often, blows, and they're trying to like get something to move for. That's mostly for hunting and foraging. You'll see them do some deep diving, so they'll arch their back and they'll go right down and they'll stay under for about 10 to 15 minutes and resurface. Especially if you see them like surfacing in like a non-directional manner. So you'll see them going one direction and then change and they coming up in odd directions. Then that's usually they either feeding, it's suspected feeding or suspected hunting. Then you can also suddenly see a fin like just coming flying at you, and that's porpoising. So they're usually hunting something at that stage, chasing. And so I've often seen, and then a penguin jumps out, and then this fin comes flying past you. And it's amazing, And then tail slaps is another thing. Sometimes they'll tail slap a penguin, which I saw the other day. It was Ella, and she like whacked the penguin out with her tail, and then she was porpoising after it.
As you can imagine, with a year of daily observations, the research assistants on Marion Island get to know the orca very well.
So the dorsal fin is the best way to ID an individual of an orca.
They do a lot of playing and a lot of biting, and so from their teeth there's rake mark injuries that happen and occur, and each one is like a fingerprint.
So for adult males, they're the easiest ones to spot because they have absolutely massive fins. Like some of them can be up to 2 meters tall. The females are a bit harder because it's really hard to differentiate between an adult female and a sub-adult male because the fins are more or less the same size. But this is where the different notches and the scarring plays a part.
And also along their bodies, some scratches, some injuries from not just killer whales, unfortunately, from boat strikes.
At the moment, I know there's a whale on the island. His name is Top Notch. And he's the only one I've been able to ID from all the images I've seen because he has a notch directly on the top of his fins. Yeah, it's very adorable. Some of them have perfectly pristine fins, no marks at all, which makes it tough when there's another individual like that as well.
Each animal has a name, and when a calf is born, they also obviously need names.
I was fortunate enough to have 4 newborns in my year, so I got to name all 4.
Orcas live up to around 60 years old, and they calf every 3 to 4 years. So it's a matter of luck whether a field assistant will be on the island for the year when a calf is born.
So my first one was Winter. His mother's, well, I think it's a he. I'm not sure yet. So to ID them, either you wait till their fins grow really large, or if they can roll over and you can see. But yeah, the mother's name is Razor, so like the sharpness of winter. And then Archer, that's Valentine's newborn. She's our oldest known, frequently seen Marion killer. And then Angel, so O'Neill's calf. And then Ember was the last one I saw.
The data that these field researchers collect is added to a longitudinal data set that is made available for future generations of scientists to analyze.
So like this project started in like the 70s basically. So since then it's been diligent work being done by birders and sealers and other killer whalers going out every single day to collect the data so that we'd have this incredibly expansive data set that so many people could utilize for so many different research opportunities.
Studying top predators like orca is not only thrilling because they are magnificent creatures, it also shines a light on ecosystem health.
Of course, it is. Like any species, they're the top predators. When something happens lower down, it'll affect them, and they're the first ones to kind of show you that there's something wrong in the system. So already we're seeing a difference in the number of killer whale observations around Marion. Each year it's been dropping drastically. So something's happening. Don't know if they're now going back to other islands or if maybe the fish stocks are moving away from Marion. So that's why we need to keep studying, keep observing, try to see what's going on.
Okay, let's turn our attention to another charismatic black and white creature. For all those who have been patiently waiting to hear about penguins, please come forward. Your time is now. Meet Dr. Azuyanewu Makado, who is the chief scientist for shore-based scientific research on the Marion 2025 voyage. He is a zoologist who specializes in seabirds, and his interests are to study how biology and oceanography intersect in the study of Marion Island's penguins. You might recognize his voice from episode 1.
I first entered the Prince Edward Island in 1999, just when I was a field assistant.
Yes, your calculations are correct. Dr. Makado has been doing research on Marion Island for more than a quarter of a century.
Macaroni and rockhopper penguins are my favorite. Although recently I've needed to focus more on king penguins.
All penguins are obviously very cute. King penguins are the second largest species of penguin, with very neat and tidy markings around their faces. Macaroni and rockhopper penguins are a bit smaller and they look quite similar. Experts like Dr. Makado refer to them as Mackies and Rockies.
The Mackies and Rockies are unique species. They symmetrical breed in this area, and we train the population in size over time.
Both species have that spiky orange eyebrow hair that makes them look a little bit like mad professors in tuxedos.
What was very interesting from what we gathered, they all, when they come back, to avoid some sort of competition, they tend to breed far away from each other. These rockhoppers normally goes to those smaller Nyana beaches with rockeries, where they will jump over those small beaches, whereas the Mackies, they go to a patchy area and try to breed there. But the other thing which we have seen, they also try and avoid the competition when they go to sea. The Mackies seem to undertake further distances from the island, whereas the Rockies will follow, but not at the same time. They will follow and then occupy a different environment, even though there's some overlaps. The Mackies dive deeper, whereas the Rockies try and remain more into the surface. The Rockies seems to like a lot of crustaceans, those that are around the islands when breeding, whereas the Mackies have a solid mixture of crustaceans and the fish. So those competitions, exclusive sort of theory that they display, even though they're breeding, they're feeding in more or less the same place, same species, and they're foraging in more or less the same place. It's quite interesting.
Over the decades, Dr. Makado and his team of field assistants have gained all these insights into the behaviors of the penguins through rigorous observations and samplings.
Although the department mandate is to look at the long-term research and monitoring, there are those smaller key drivers of change that we tend to want to understand why this is happening, why that is happening, and that's when we started with this to understand more of what is their behavioral response to certain episodic events and long-term events as well.
Let's turn our attention to the seals of Marion Island.
Yeah, so in the past, the sealer used to be one of those rough guys dropped off on an island, killing seals and chopping them up and boiling their blubber for oil, for the oil industry, for lighting the cities back in Europe a long time ago. But these days, the sealer is more of a conservation kind of guy. Goes there and he censuses seals, he checks health and population growth of seals on whatever species he's working on or whatever island he's working on.
Like those studying the killer whales and penguins and birds, every year overwintering field assistants gather valuable data about the elephant seals and Antarctic fur seals that live and breed on the rocky beaches of this island.
Yeah, toughest job on the island.
Here is Dylan Manyoka, a returning overwinterer, explaining one key part of the job, doing a seal census.
You probably on the island at base on 3-day intervals, because our cycle is 10-day cycles spread across 2 stretches. So we have a north stretch and then we had a south stretch. And then in a day, what would basically happen is that we'd split those stretches into days of censusing, where we just go and count all the animals on the beaches, how many are tagged and how many are untagged. And the most stretch would be split into 2. So that can be done over a 2 days period, and then south census is split into 3, and it was done over a 3-day period. And along that time period, we have so much other things that we have to work on to contribute, because we also have people who are doing population genetics on the elephant seals. We also have people who are doing stable isotopes on them.
As an example of the genomic analysis that is done with this material, here is Yinhla Shiklomuhle, another Marion Island veteran who is currently doing his PhD on seal paternity patterns.
So I'm working on population genetics and paternity of southern elephant seals from Marion Island. In breeding season, one bull normally controls a harem of like between 1 to 8 cows, and then there are subordinate bulls that are trying to mate with their females. But then because they are subordinate, they get chased away. So I want to confirm if the alpha male is the one who's actually fathering all the pups that born the following year. In the past, they used to deploy tracking devices on elephant seals, and then some of them, they happen to be at sea during breeding season. So one of my questions is to check who are they mating with when they are at sea, to check their paternity.
In addition to counting, sealers have to take biopsy samples for studies on seal population genetics. That means getting a bit of organic matter from the animals by clipping a bit off a hind flipper. Dylan promises that it only feels like a pinprick to them.
They're quite hardy, so you would snip a little piece off of them and then put it in a jar. But there's a new option in collecting mold samples, and that's actually much easier and much nicer, because most of the time you don't always see the animals during molt, so the biopsying actually accounts for that. But most of the animals do return to molt. You even get animals that you'll never see throughout the year that only come out to molt. So it's very important to collect those samples, and so you just go to the animal, especially if it's calm and safe, and you just peel it off, and it's therapeutic to them and to yourself.
As well as the glamorous work of counting seals, and perhaps even more glamorous work of peeling off bits of their itchy, dry skin when they molt.
You just peel it off. They like it quite very much.
Sealers also have to tag seals.
Tagging is actually the long-term monitoring, which has been done now for 42 years. And the group we've left will be the 43rd group. So we have color-coded cohorts to identify which animals were born in which particular year. So during breeding season, as soon as the pups are born, the first thing we attempt to do is we try and tag as many pups that have tagged moms as possible. So as breeding season proceeds, there comes a time when the pups wean. And that's our opportunity, because their skin is now tougher. That's also the safest time. They're no longer being protected by their moms when they sort of get banished from the harem. So they're on the outskirts, so it's safe to tag them. And they're not quite as bright as the other animals, because it's their first time into the world. They were dropped there, and then their mom just abandons them at one day. So they don't know what to do. So they just hang around. They're just so like colorful, and they don't know what's going on when you tag them. It's probably one of the most dangerous jobs. So the flesh between your thumb and your index finger, we would call it your outer interdigital, and that's where we would place our super smalls. And then the jumbos would be between your index finger and your middle finger.
All of this adds to longitudinal data sets about each generation of seal on Marion Island.
They're very charismatic, and they have personalities. They're closer to humans than people think.
Dylan makes it sound really fun, but it's tough work. And of course, the sealers have to be really careful at all times, not only to protect the animals, but also to protect themselves.
You get the very calm ones, get the very aggressive ones. You even get personality clashes. There are seals that just don't like you for a particular reason, but they like your partner. So yeah, they'll always chase you and then leave the person you're with alone. I feel like they're very in touch with their emotions.
The sealing teams also have to weigh the pups at a particular point in the year. Can you think of a more fun job than weighing a seal pup?
To weigh a wiener, they're quite chunky little things, so it takes 2 people to weigh one.
Here's Camilla Smythe, another sealer, explaining how they weigh the chunky pups.
We have to put them into a net, and then there's a big metal pole, and one person stands on each side, and you lift the pole up, and there's a scale in the middle. The fur seals that we'll be weighing them as pups from one of the species from when they're newborn, one of them from when they're a little bit older. And those you can pick them up by the flippers gently, and you just dangle them from a scale, so you can do that with one arm.
We have a rope that is tied into a bit of a knot, and we have a little small scale. So you chase them, and then you grab them by the hind flippers, and then you have the rope around the little flipper, and then you just hang them upside down for a bit, and then you read the scale, and you drop them, and in the process you just quickly check their gender. So it doesn't really harm them.
Elephant seals are really huge and intimidating animals, and each beach is dominated by a beach master, the male in charge of the community of seals on that beach. The sealers have to be really careful and pay attention to all the social hierarchies on each beach while they do their work. Here is Kwanele Kuzwayo, another sealer, weighing in.
There is also a method just to sort of estimate the weight of a female or males. We just take picture right around that seal and ensure that it doesn't move. And then we use a certain model to estimate the weight of the seal. So even if it's a beach master, which is the seal that all other seals are scared of, we need to do that. So it's a scary job, but yeah.
Yeah, you probably will sustain more harm than them. They're feisty little animals. So yeah, a lot of people have gotten bites, but people are proud of their bites. They battle scars.
It's very physical work, but Marion Island is one of the most beautiful places on planet Earth. And all the field assistants working there are gaining valuable experience in the practicalities of gathering data for biological and zoological research.
It can be a pretty dirty job. A lot of the seals are amongst the bird colonies, so it's a lot of guano everywhere on the seals, on us.
That's a thing people tend to forget when they hear that we work in the field. They think, oh, it's so cute, you're touching the birds or picking up the pups. And they don't realize how absolutely disgusting it can be. Because animals are filthy.
For this episode, I'll leave you there, imagining yourself among the smelly guano-littered beach full of chunky seal pups, trying to figure out how to get the tag on the right place on the flipper without upsetting its mum, or how best to pick it up and weigh it without upsetting it for more than a couple of minutes. Think also about how many careers in science have been started on this island. Young graduates come out as field assistants, and years later, they've earned their master's degrees, their PhDs, and become experts in their field, contributing pieces to solving the great puzzle of biodiversity on planet Earth. As Marion Island is a key breeding site for penguins and seals and orcas, the oceans around the island are a really key study site for getting deeper insight into plankton populations and then in turn the health of the oceans. There are just so many creatures, so many that we haven't even been able to touch on, that are being studied in this special piece of the Southern Ocean by a diverse group of very proudly South African scientists. I hope these glimpses into their work, their research questions, and how they collect their data has been inspiring, and that you're motivated to find out a bit more about their research. We'll drop a few relevant links into the show notes for this episode if you want to explore more. In the next and final episode of this special SA Agulhas 2 Marion Island voyage season, we'll think about how everything links together. Plants, animals, land, sea, climate, islands, training, policy, the ship, the island, the planet. We'll also consider what the sub-Antarctic science program means for South Africa and future generations of scientists. Stay tuned. My voyage mates on the SA Agulhas 2 from the Marion 2025 takeover voyage still have a lot to tell you about their work. And there is still a lot to find out about Marion Island, including a little bit of the unexpected. For example, did you know that there is also a little bit of experimental space research taking place there? You'll have to tune in to the 4th and final episode of the season to find out. This podcast was created, recorded, and produced by me, Mahita Ikani, with stellar production assistance from Andrew Burnett, who also did all the sound editing and sound design. Thank you, Andrew. My berth on the SA Agulhas 2 for the 2025 Marion Relief Voyage was provided by the SANAP Artist-Writers Project, directed by Professors Sean Lavery and Jean Brundrit, who were both absolute stars in letting me join the voyage as their representative at the last minute and helping me get my head straight before I walked up that gangway. Big thank you also to Dr. Adrian van Eerden-Wharton for helping me get all my ducks in a row. She was the artist in residence on the ship in 2024. Big thank yous to all the guests who appear on this episode. Emma Swanepoel and Sarah Duprier, both MSc students at the University of Cape Town. Dr. Zimkita Gebe, Marcel van den Berg, Dr. Azwianewu Makado, all from the DFFE. Chandler Patel, Megan Clarkson, Dylan Manyoka, Yinkla Shiklomuchle, Andile Kuzwayo, Yana Rao, Camilla Smythe, Juanele Kuzwayo, and Peter Cunningham from SAPRI. The production of this podcast was funded by the South African Research Chair in Science Communication, which is funded by the South African National Research Foundation. I'm the humble and proud shareholder of that SACI. The distribution and marketing of The Academic Citizen is done by Fumani Dwaha. Huge thanks to the captain, officers, and crew of the SA Agulhas 2 for keeping us all safe on the voyage. To find out more about the South African National Antarctic Program, the ship, and the islands, check out www.sanap.ac.za. A reminder that all of our guests on this episode were speaking in their personal capacities and are not officially representing any of the organizations mentioned.